cells that are matured in the gene EVA. Further, the analysis shown that EVA mRNA in the matured cells is the 1.8 kb in the length, versus in the mRNA from the wild type, even is the 1.2 kb as a length. However, the genomic sequence of the mutation, even the gene only shows the signal different from the wild type. The mutation is not the promoter. What is the most likely explaining? I think this is explaining there is an interaction mutation in the genes. This is based on a different gene. I think the first one was the answer. Question 9 says, Tom is a male colorblind and has a hemophilia B. Mary is a female, not colorblind, and she doesn't have the hemophilia B. Both hemophilia B and colorblind X-linked recessive. They have the daughter. The first has the hemophilia B, but normal vision. The second is a colorblind, but doesn't have hemophilia B. Third has both colorblind and hemophilia B. What complete explanation account for this phenotype of the three daughters? So this one is, I think, Mary was a carrier of both diseases. The first daughter got the combination of the X, in which is a color-blinded allele, was lost due to crossover. Similarly, the second daughter in the X chromosome, which is hemophilia B yellow, was lost. Question 10. Deep sea environment unlikely lack oxygen but contained lots of the phosphorus and the sulfur. As a consequences of the environment difference, what has made you predict that what would be major difference in the ETC between the surface organism and organism from the deep sea environment? I think deep sea organism will see the phosphorus to make the ATP-VA ATC. Okay, right now I am putting the computer away and then I am taking the answer for this question. So for question 11, I put the red phosphocoprotein in the membrane of the cells. Normally having no fluorescence, but now you see in the blue light and will they show the fluorescence. I activated the RPA protein in the area of the I cell and then measured the amount of the fluorescence in the area B, the cell. Data from the two different cells is showing the graphs based on the data. Assume the equal initial activation of the RPA in the two cells. Most likely, which cell has a saturated in the cell membrane? I think again in the cell B is a protein differs least rapidly in the cells, indicator lower the membrane liquid in the cell. Protein ionophores created the pores in the cell membrane, which allow to roughly diffuse the across the cell membrane. The pH is a 7.2 versus within the cells 4.5. I add the protein ionophores in the cell. Most likely what happened to protein Y? Yeah, I think the protein will diffuse from the outside to inside, allowing the concentration of the gradient. Transposase is the element within the genome that can randomly jumping from the location of the genes to another, such as jumping of the cells, mutation with the genes. During with the cells division, a stage made the such jumping beneficial to the species.